So you're telling me that you can't take mRNA and turn it back into DNA? Can I speak to the manager?
You talking to me?
DNA is our genetic code. It has all the instructions for our cells. So DNA's role, one, is that it keeps information, okay? It also has to be able to copy information because when our cells divide, we need each daughter cell to get a set of DNA. And it passes on information. That's how we pass on information through generations. So this is what a small piece of DNA would look like. We call it a twisted helix because it has this, it's like a ladder that's been twisted up. So DNA is made up of small building blocks or monomers. So each one of these little parts is a building block of DNA, and that is called a nucleotide. So nucleotides are the building blocks or monomers of DNA. So now I've zoomed us in further to see what one of those nucleotides is made up of. And it is made up of 3 different pieces. It's made up of a phosphate group, and it's made up of a sugar. Now, in the case of DNA, that sugar is called deoxyribose. Sorry, that's a parenthesis. Deoxyribose. And that's why what DNA stands for is deoxyribonucleic acid. So, and then this last part, which we'll talk more about in just a second, is a nitrogen base. That's an O. Now remember, we have our whole DNA molecule here. We zoomed in to look at just one nucleotide. And now, if we look at just the nitrogen base of that nucleotide, there are 4 different possibilities. So these 4 are the different nitrogen bases that could be in that nucleotide. So we have guanine, adenine, cytosine, and thymine. Those are our 4 nitrogen bases. So based on what we've learned so far, here is what one strand or one side of that DNA ladder would look like. We have a nucleotide here. And remember, each of these nucleotides is made up of a phosphate group, a sugar, and a nitrogen base. And you can see that we can have different nitrogen bases, one of those 4, on each nucleotide. But how do we get from just this one side of the ladder, 1/2, to these 2 sides paired together? Well, that's what we're going to look at next. So to figure out our other strand of DNA, we use what's called the base pairing rules. And those are that adenine always pairs with thymine. So A will always pair with T. And guanine always pairs with cytosine. So C always pairs with G. Okay, so let's put everything that we've learned together. First, let's try those base pairing rules. So A, since we have adenine here, that means over here we would have a thymine. G pairs with C, so we have a cytosine here. Since there's a thymine there, we would have adenine here. Cytosine pairs with guanine, and guanine pairs with cytosine. So that's how you use the base pairing rules to figure out the second strand. Now let's draw in everything that we've learned, because we've learned a lot of terms. One of these are the building blocks, which are called nucleotides. The nucleotide, each nucleotide has 3 parts. We have our phosphate group. There is the sugar, deoxyribose, and in here is the nitrogen base, which could be either A, T, G, or C. Then this whole side of DNA, each of these are called the backbone, or sometimes you'll hear it called the sugar phosphate backbone because it's made up of sugar and phosphate. So that's the backbone. Now, what bonds are holding these together? So let me move over to this side where there's more room. The bonds that are holding the backbone together, these are called covalent bonds, and we learned about those at the very beginning of the school year. We learned that in covalent bonds, they share electrons. And then these bonds that hold the two sides of the ladder together, those are called hydrogen bonds. So this brings together everything we've learned about the structure of DNA. So make sure that you label these things in your notes so that you have it to look back on. And now you've learned the structure of DNA, and we'll get to practice it this week.